Morning, Doctor.
Okay, morning. Assalamualaikum and good morning, everyone. So can you all hear me?
Yes.
Yes, doctor.
Okay. So today's lecture will be on muscles and muscle tissue. So I'll do both lectures together because this one is very short. So I'll finish both lectures by 10 a.m. lah, inshallah. So you have some time after that to prepare for your afternoon session. Lah. Okay. So introduction to myology. Okay, as I mentioned earlier, for osteo in anatomy or in medicine, osteo would refer to bone, chondro will refer to cartilage. Okay, so myo will refer to muscles. In a lot of you will come across a lot of myo in the future, and that will be referring to muscles. Okay, so in this lecture, you will describe the basic characteristics of muscles, the parts and attachments of skeletal muscles, the nomenclature, so how we name the muscles, describe some arrangement of muscle fibers, and also the grading of muscle power. So by definition, myology is the study of structure, arrangement, and action of muscles. So basically, you're studying the anatomy of the muscles. Derivation of the name comes from the word mouse. Okay, because a lot of the muscles, the shape resemble a mouse, where the tendon of the muscles look like the tail of the mouse. So it is a contractile tissue which causes or brings about movement, and it is the motor part of the body. So characteristics of muscle. First, it has the ability to respond by producing action to by responding to impulse created by action potentials by nerve fibers. So this is called excitability. So next is contractility. Okay, so it can contract in response to the impulse. Extensibility, it can be stretched, and elasticity, elastic like the rubber band, so it can return to its original shape and length once after the stretching. Okay, so in terms of morphology of the skeletal muscle, it has a muscle belly. So the pink or red orange area here are all the muscle belly, and the white bits here are the tendons or aponeurosis lah. In certain areas they have aponeurosis. Aponeurosis is a thin like sheet which I will show you the picture in a bit. Okay, but in most muscles they have tendons. Okay, this is what helps to attach the muscle to bones. Okay, classification, we have a lot of classification, but for introduction, we have long muscles, short muscles, broad and orbicular muscles. Orbicular are like circular shapes. This is a flat tendon. Is a type of tendon called the aponeurosis. Okay, it's like a thin sheet. Okay, so here at your abdomen, for example, you have this aponeurosis. It is attached to the external oblique muscle of your abdomen. Okay, so each muscle has an origin and insertion. So the origin is the fixed attachment. The insertion is the part where is movable. So in terms of action, we have agonists, antagonists, synergists, and fixators. And for naming purposes, we name the muscles according to the shape, the size, location, and their points of attachment. So if naming the muscles according to the external muscle shape, for example, the fusiform muscles is the biceps, the parallel muscle is the sartorius. I think for now it's more of an introduction. In your musculoskeletal system, you will learn more about what the muscles are and so on lah. For now, you just need to know there are different shapes of muscle. According to the shape, is how we give the name. Okay, so for example, I think in this picture is clearer lah because you can see the shape kan. Okay, so for example here, this is a fusiform shaped muscle, which is the biceps brachii, and rectus abdominis is a parallel muscle. You can see the muscles running parallel with each other, and pec major is a convergent muscle. You can see that the fibers converging into one tendon. Unipennate meaning feather-like. Pennate is like feather-like shape. Okay, so unipennate means there's one feather-like shape, which is this palmar interosseous. Okay, bipennate muscle meaning two feather-like projections coming out from the tendon. Multipennate means a lot of feather-like, feather like from the bird's feather tu. Okay, this is what it looks like. So multipennate is feather-like, a lot of feather-like projections of the fibers. Okay, and in your orbicularis oculi or your eyelids is circular shape. Okay, so next to the muscle or close by to the muscle, we have accessory structures or other structures. Remember my osteology lectures or joint lectures as well. We have other structures apart from the bone or the joints itself, right? Okay, we learned about bursa there as well in the joint lecture. Okay, so because joint muscles and bones are close together, so they are accessory structures which are close by with each other lah. Okay, so here for example, in the hip bursa, we have trochanteric bursa at the greater trochanter, ellipsoas, and gluteus medius bursa. Don't worry, we won't ask you all these names. But just to show you that we have accessory structures near the muscles lah. Okay, and we have fascia, which is like a thin covering. Remember in bone we have the periosteum, right, covering the bone. Okay, it's like a protective layer. Okay, so in muscle we have the fascia, which is deep and superficial fascia. So two layers of fascia covering the muscle. Okay. So we also have a tendinous sheath, which covers the tendon and protects the tendon. So it permits the tendon to stretch and not adhere to the surrounding fascia. There is two layers, the synovial sheath and the fibrous tendon sheath. As the name suggests, the synovial layer contains the synovial fluid. Okay, and the fibrous layer is the outer layer lah. Okay, so if you imagine here, this is the tendon inside, okay, and this is bone of your finger. Okay, so you can see here, these are all tendons from muscles. The muscles will be in your forearm, and the tendons of the muscles goes to your digits, fingers. Okay, and they are covered by this tendinous sheath, which is in purple. Okay, as a protection. Okay, and if you do a cross section here, you will see this is what it looks like. So this is the tendon with the surrounding tendinous sheath. Okay, and this is inner synovial layer with the cavity for the synovial fluid, and this is the outer fibrous layer. Okay, so these tendons or small tendons are protected by this tendinous sheath. So function of a muscle. Muscles can act as the agonist, antagonist, synergist, main principal muscle, or auxiliary or accessory muscle. So for example, we'll see the example here. Okay, so in when you are lifting a glass of water, for example, so your biceps brachii is the prime mover, because the biceps brachii creates the muscle, the action of elbow flexion. Okay, and the brachioradial.
Hello guys, can you hear the doctor?
Doctor.
No,
It's muted.
I think she accidentally muted herself.
Doctor, we can't hear you, doctor. Sorry.
Hello?
Yes.
Okay, now we can hear.
Okay, sorry. Which slide did you stop hearing me? I will
In
repeat.
the
The
brachioradialis.
brachia,
This one,
the
huh?
bicep.
This one.
No, the one before this one.
The one before this one.
No,
This
the
one,
one after
function.
it. The one
This
after
one.
it. This
Yes.
one, yes.
You're explaining about the glass of water.
Okay. Okay, I'll repeat this slide lah. Okay. So basically, oops. So for example, when you flex the elbow, the biceps brachii is your main muscle that flexes the elbow, which is called the prime mover lah. Okay, so there are two other muscles that also help to flex the elbow, which are your brachioradialis and brachialis. So these are termed as synergist muscles, which help the brachialis. And if you it's not seen here, but you have another muscle at the back of the arm here, and that will be your triceps, not shown in the picture, and that will act as an antagonist because it acts when the biceps contract, the triceps will relax. Okay, so that the action is opposite of the prime mover, so the triceps is termed as the antagonist. Okay.
Excuse me, madam.
Yes.
Can you go back to the previous slide? I want to ask something. Okay, the brachialis and the biceps brachii, are they the same or different?
No, different.
Different, okay.
Yeah,
Thank
if you see here the biceps brachii, if you cut the biceps brachii, you will find the brachialis inside. It's more deep. Yeah. Don't worry too much about the muscle names, like the muscle itself, because you will learn in more detail during your musculoskeletal system. But for now, you need to understand the concept of agonist, antagonist, synergist, and so on lah. Okay.
Okay, thank you.
Okay. So function of a muscle, apart from contracting and agonist, antagonist, synergist, and so on. Okay, so it also acts as a fixative or stabilizing. It can act as a something that fixes something lah. Okay, and some muscles are neutralizing muscles as well, and some muscles are one jointed muscles. They only cause one movement, and some are double jointed or multiple jointed. Okay, so what this means is basically how many joints that the muscle crosses over. So if it crosses over one joint, then it's called a one jointed muscle. If it's double or multiple joint, it's named accordingly lah, double jointed or multiple jointed. Okay, so for example here, if the you can see the brachioradialis, it crosses the elbow joint, which is one joint, so we call it a one jointed muscle. Okay, so function of a muscle, part 3. Okay, so we have flexors, extensors, adductor, abductor, rotator, levator, depressor, pronator, supinator, opponents, sphincter, dilator. So we have a lot of muscles that are named according to their action. So for example, in the forearm, all your flexor muscles are named, they have the name flexor at the end, most of them. So and adductors, example, in your thigh, you have your adductor pollicis and adductor magnus and so on. So they are named sometimes according to how they function lah. Okay, and sphincters, you have at your eyes, for example, you have sphincters and dilators as well. Okay, so they also muscles also help to maintain tone. Okay, and also postural muscles help the posture of the body. So for example, muscles that are at your back muscles, they help to maintain your posture of the body lah. Okay, so this is for function of muscles. So what are innervations of muscles? So they have neurovascular hilus. Okay, neurovascular, neuro means nervous tissue or nerves. Vascular means blood vessels lah. So you always see a neurovascular in anatomy teaching. Okay, so hilus is just an area lah, or an area. So for example here, this is the neurovascular hilus. So it is the neurovascular hilus is the point of entry for nerves and vessels into each muscle. Okay, so if it's a diploneural muscle, it is innervated by 2 nerves. Plurineural, it is innervated by many nerves. Okay, and as for neural fibers that supply the muscle, there are 3 different types. Some give motor innervation, some give sensory innervation, and some give autonomic innervation. So autonomic, for example, the muscles in your GI tract. So you cannot control your peristalsis, right? So that is autonomic innervation because it is automatically controlled by your brain. However, for motor, for example, your biceps and so on, all your skeletal muscles that you control for movement, that is your motor innervation lah. Okay, and sensory innervation, it's your sensory lah. So if you feel hot, cold, pain and so on, they are innervated by sensory muscles. So motor innervation are by alpha motor neurons. Okay, they are from the anterior spinal horn. We also have neuromuscular synapse at the near the motor. This just means neuro means nervous system or nerves, muscular means muscle lah. So the synapse is the junction where the nerve meets the muscles, and that's called the neuromuscular synapse. It's a termination of axon at the surface of the myofiber or the muscle lah. Okay, so what is a motor unit? A motor unit is a group of myofibers innervated by the same motor neuron. Okay, so a motor unit creates simple, rough movements. A big motor unit will create so a big motor unit creates simple and rough movements, but a small, precise motor units create a more delicate movement. So we'll see here. Okay, I'll explain more about the motor unit in a bit lah. Okay, so we'll just talk a bit about muscle spindles first. Okay, so muscle also has sensory detectors, which are muscle spindles. Okay, so it helps in proprioception. So proprioception is when you are aware of where your position of the body parts is. So for example, if I were to ask you to close your eyes and I move your finger downwards, you will still be able to tell me that I've moved your finger down without seeing me do it because you have the perception of where the position of your body parts are, for example. So that is called proprioception. Okay, and this muscle spindle give feedback of the position of your body parts, okay, through the receptors and nervous system lah. Okay, so it is an important part of your muscle. So it consists of several myofibers surrounded by fibrous capsule innervated by fibers of gamma motor neurons. Okay, it also detects the difference of grade of contraction of extra and intrafusal fibers. Okay, I'll talk about that also in a bit. Okay, so for naming purposes as well, we have, for example here, muscles that are named according to the origin and insertion. So the sternocleidomastoid, the sterno means the sternum, so it originates from the sternum. Okay, here this is the sternum. Okay, cleidomastoid. Cleido means clavicle, so this is the clavicle, and mastoid is the mastoid process of the bone. So this muscle here is named mastoid because of the origin and insertion. Okay, and so the origin and insertion, as I mentioned earlier, origin is the point that the muscle is fixed and does not move. Insertion is where movement occurs lah. So for example here, the biceps flexes the elbow, so the flexion occurs near the insertion and the origin is fixed. This part of the biceps does not move. Okay, so vascular supply of a muscle. We have the neurovascular hilus, which I mentioned in the how the nervous system goes into the muscle or supplies the muscle. Okay, the same hilus also creates entrance and exit for the vessels. So the flowing blood volume, of course, depends on the activity of the muscle. The more active the muscle, the more blood needs to go in and supply oxygen. Okay, the tendons, however, receive poor blood supply. They have their own blood supply and lymph vessels coming from branches from vessels supplying the muscles, periosteum, and tendons. Okay, so growth and regeneration of muscle in terms of length. The myofibers at their ends do grow in length. In terms of width, they also grow in width. From birth, however, the amount of myofibers does not change. So they are not replaced, but they just grow in length and width. Okay, no regeneration or renewal of cells like the bone lah. Okay, so we have also structures surrounding each muscle fiber here. So we know that there's fascia surrounding the outermost layer. Okay, so that is also called perimysium externum. And we have osteofacial septum, which is fascia, but against each muscle fiber, surrounding each muscle fiber. Or we call muscle cells muscle fiber because it's bigger than your normal cell. So when I say muscle fiber, I'm referring to one muscle cell like this lah. Okay. So because of the fascia, we have this fascia. So there is a syndrome called Compartment Syndrome where there is leakage of blood or any type of injury can cause leakage of leaking into this area lah. Okay, and this is called compartment syndrome because this is a small enclosed space. If there is blood accumulating inside, for example, it can cause compression of the vessels and finally ischemia with possible necrosis. So this is a common side effect when there is any injury or fracture of the bones, of the long bones of the thigh, leg, arm or forearm, because the injury or the blood from this injury leaks into this space. This space, osteofascial space, and it compresses these structures here. So you can see here, this is your blood vessel, your vein, and also your lymph node or lymph vessels. Okay, so when blood leaks inside here, okay, so this is bone. So if there's injury here and there's blood leaking into this area, so it can compress these structures, and this is called the compartment syndrome lah. Okay, so treatment is you have to surgically cut open the skin to release the pressure and release the blood out lah. Okay, so Volkmann's contraction is one example of compartment syndrome, where they get contracture of the hands and fingers because of the degeneration, meaning because of the compression, the part of the muscle belly does not receive enough blood, and they get this contracture lah, because they don't get blood supply. They contract without, they cannot function properly lah. Okay, so this is just an example. You don't really need to know the details for now. Just understand that if you relate the anatomy, which is they have this fascia, the osteofascial fascia and the fascia outside. Okay, because of this this fascia has these structures inside. So if there is leakage of blood inside this area, you can get injury or compression of the vessels and causing ischemia lah. Okay, so just to relate the anatomy with the disease. Okay, and in terms of muscle tests as well, so if you usually we use this in patients who come in with muscle weakness. For example, in stroke, in patients with stroke, okay, you need to assess how severe is the impact of the disease or the stroke. So you will do a test to see whether the muscle and give grading to the muscle power lah. So, for example, if the patient with stroke, for example, comes in and say they have weakness in the arm, okay, they cannot move the right arm, or they feel it's a bit heavy or weak. Okay, so you do this test on both arms, and you see, you can grade the power according to the reaction that you find during examination of the arm. So, if you ask the patient to, for example, flex the biceps, or use the biceps to flex the elbow, okay, and you don't see any signs of contraction, no movement at all, okay, no movement or flickering anything at the arm, so you grade the patient's arm as 0 grade, meaning there's no sign of contraction at all. If you ask the patient to move the arm or flex the elbow, and you see some twitching, but the hand does not flex, just some twitching near the biceps area, then you give 1 mark, 1 grade. Okay, if you ask the patient, the patient move a bit but doesn't overcome the resistance, meaning you ask the patient to move against your hand, for example, very weak movement, that would be 2. If the patient can move against gravity, meaning you ask the patient to lift the hand away from the bed, for example, or away from a table or something like that, upwards, and then you can give a 3. Okay, if you ask the patient to do the same thing but you put resistance, and the patient can overcome a bit of resistance, then you can put 4. If the patient can do what you can do, that would be normal or grade 5. Okay, so I think in this easier to remember 0, 3 and 5, and then all the others will be in between lah. So if there's no sign of contraction at all, you give 0. If they can overcome gravity, you give 3. If it's normal good function, you give 5 lah. Okay, so if it's in between that, you give 4. If it's in between weak and no signs of contraction, you can give 1 or 2 lah. Okay, so just twitching, not actually moving the part, you give 1. If there's slight movement of the arm, you can give 2. If more movement, they can lift the area against gravity, 3. They can resist a bit, 4. Resist a lot, 5. So normal function lah. Okay, so another clinical correlate, or that something that you can correlate the anatomy with the clinical side is rigor mortis. It is actually the state of muscle rigidity that begins 3 to 4 hours after death and lasts about 24 hours after death lah. Okay, so what happens after death is that the calcium ions leave the sarcoplasmic reticulum and enable the myosin heads to bond to the actin fibers. So basically in the muscles, once death occurs, the calcium will go into the blood system lah. Okay, and causes the myosin and actin to bond with each other. Okay, so this causes the muscle rigidity, or all muscles to contract. Okay, so when all muscles contract, that's why the body will be found very rigid 3 to 4 hours after death. This is also used in forensics to determine the time of death. If they don't know what time the patient or the okay, so they can use this as a clue lah. If the patient is still rigid, like all the muscles are contracting, it's an early, they just died for how many hours. But after 24 hours, okay, all this the myosin and actin will stop to bond. Okay, the bonds will start breaking. Okay, because the proteolytic enzymes will start to eat up the proteins lah, which are the myosin heads and actin fibers. Okay, that will be about 24 hours lah. Okay, and then all the muscles will start to relax. Okay, they won't be rigid anymore. Okay, and that's how you tell if the patient, the muscles are relaxed instead of rigid, then you know it's already been more than 24 hours after time of death lah. Okay, so and sometimes that's why when you hear stories saying that, you know, suddenly the cadaver moved. Okay, so they don't really move per se, but actually this action lah, okay, because they were rigid before due to the myosin and actin bonding. Okay, but after 24 hours, the proteolytic enzymes have eaten up the myosin and actin. Okay, so the muscles start to relax, so they suddenly kind of move. Okay, so that's called rigor mortis. Okay, so I'll continue with the second lecture because it's related to this. Okay, oops, sorry. Okay, so this is learning about muscle tissue. So function, I think we talked a bit about the general function of muscles just now, but muscle tissue is looking at the microscopic part of the muscle. Okay, it generates movement, stabilizes body position, also control volume of the organs, for example, muscles with sphincters. Okay, it helps with motion of the substances in the body. For example, your blood vessels, they have muscles as well to pump blood into different parts of the body, and we have muscles that generate heat. Usually, skeletal muscle. If you when you shiver, that is from muscles that help to generate body heat. Okay, so there's 3 types of muscle tissue. We have smooth muscle tissue, striated or skeletal muscle tissue, and cardiac muscle tissue. So smooth muscle tissue is found in your involuntary contraction areas lah. For example, in your blood vessels. Okay, you can't control how the blood vessels move the blood, right? Okay, so it's involuntary control. And also your GI tract, you cannot control peristalsis. Okay, so that's your smooth muscle tissue lah. Okay, your skeletal muscles make up the most muscles in your body. So skeletal, as the name suggests, it attaches to bone. Okay, and helps with body movement. And cardiac muscle is found in your heart. Okay, for muscles, if you go to the cell level, as I mentioned, the muscle cells are called muscle fibers. Okay, and they have special terms for their different parts as well. So the plasma membrane is called sarcolemma, the cytoplasm is called sarcoplasm, the smooth endoplasmic reticulum is called sarcoplasmic reticulum, and the mitochondria are called sarcosomes. Okay, so organization, they are muscle fibers that are supported by connective tissue framework. I think I mentioned the fascia and so on earlier. Okay, so they have tendons and aponeurosis as well, and they carry blood vessels and nerves as well. Okay, so just to show you the arrangement of muscle fibers. So if you take a muscle here, so for example, this is your pelvis and this is your humerus. So this is your is this? Yeah, this is your pelvis. Okay, so this is a muscle attached to your femur. So if we take one section, okay, and you do a cross section or a cut through the muscle here, you will find that it is made of many muscle fiber bundles. Okay, so the muscle is not just made of many cells, but it is made of layers and layers and layers of cells. Okay, so if you cut a muscle, you will find many muscle fiber bundles. Okay, muscle fiber bundle. Okay, and if you take one muscle fiber bundle and you cut that, you will find it is made of many muscle fibers. So these are all many muscle fibers. Okay, so this is one bundle, this is one bundle, and these are the muscle fiber bundles. Okay, and if you take one muscle fiber, okay, you will see it is made of many myofibrils. So this is a myofibril, and if you take one myofibril, it consists of many myofilaments. So there are many layers of structures in the muscle fiber that make up the muscle fiber. Okay, and the muscle fiber is the cell. Okay, the muscle cell. These are smaller units inside the cell. Okay, so if you focus on one muscle fiber or muscle cell, you will see it has its own plasma membrane, which is called the sarcolemma, its own endoplasmic reticulum, which is the sarcoplasmic reticulum, mitochondria, and myofibrils. Okay, it has these T tubules, which we will talk about in a second lah. Okay, so it is also covered by epimysium, perimysium, and endomysium. So like the covering of your bone, as I mentioned, it has periosteum. Okay, so muscle also has covering called epimysium, perimysium, and endomysium. Okay, I'll show you. Okay, so if you again take a muscle like this, so the epimysium surrounds the whole muscle, the perimysium surrounds one bundle, and the endomysium surrounds each muscle fiber. Okay, so you have 3 layers of mysium. Again here, the epimysium covers the whole bulk or the whole muscle, and if you take one bundle, one muscle fiber bundle here, it is covered by perimysium. Okay, and if you cut the muscle fiber bundle and you take one muscle fiber, it is covered by endomysium. Okay, so if you look or focus on the muscle fiber or the cell, okay, each muscle fiber is elongated and unbranched. It has many flat nuclei located beneath the sarcolemma. You can also see bands of light and dark bands, which are A and I or H and I bands with Z line intersecting the I band. Okay, this is we will show you in a bit lah why we have these dark and light bands made from the myofibrils. Okay, and the distance between two Z lines here is called the sarcomere. So this is the contractile unit, or the part that contracts in the muscle fiber lah. So arrangement of the myofilaments in the sarcomere, it consists of thick and thin filaments. The thick filaments are mainly made of myosin, and this makes up the A or H band. The thin filaments consist of actin and also tropomyosin and troponin, and this occupies the I band, but the main component is actin lah. Okay, so myosin occupies the A band, and it's a thick filament. The thin filament is actin and occupies the I band lah. Okay, so one end of each thin filament runs between and parallel to the thick ones, and I think if I show you the picture, is easier lah. Okay, so again, this is the arrangement. So if you imagine we take one muscle fiber like this, okay, and we will see this all this lah. Okay, if you focus on one muscle fiber, you will find it is made of many myofibrils. Okay, and here you can see the Z line. Okay, this is the Z line, and here is the thick. You can see here thick filament and thin filament. The thick filament consists of mainly myosin. The thin is actin. Okay, and if you see here, so where they overlap, where the thick and thin filaments overlap here, it is dark colored, and this is the H band or A band. And where they do not overlap here, coincides with the I band, which is a more lighter shade lah. Okay, so the light part here would be your I band. It is due to the thin filament. Okay, and your dark area here would be your H or A band due to the overlap of the thick and thin filaments. Okay, and if you notice here in the I band is where you find your Z line lah. Okay, and as we mentioned here, okay, the distance between two Z lines is the sarcomere. So if you imagine from here to here is one sarcomere or one contractile unit of the muscle fiber. So from here to here is one contractile unit. from here to here, or it's called a sarcomere. This is one sarcomere. Okay, so during contraction of the muscle, and actually there is no shortening of any actin or myosin. Okay, however, there is an increase in the degree of overlap between the filaments, and this is called the sliding filament theory. So instead of the actual shortening of the filaments, they slide over each other, causing the Z bands or Z lines to come closer together. So if you see here, okay, this is your thick filament, which is myosin, and this is your thin actin filament. Okay, and this is your Z line. Okay, so from one Z to another is one sarcomere, right? Okay, and during contraction, these bands do not change in size. What happens is they slide over each other closer together lah, making the Z lines closer together. Okay, so remember this, in a contraction there are no shortening of the actin or filament, actin or myosin filaments. However, there is sliding of the two filaments lah. Okay, and this is called the sliding filament theory. This one, okay. Another structure that I mentioned before is the transverse T tubules. Okay, it is special for the muscle. Okay, so as the name suggests, it is T-shaped. Okay, and it lies transverse inside the sarcolemma. Okay, so the function of the T tubules is to conduct impulse from the surface of the cell, which is the sarcolemma, down into the cell and specifically to another cell in the cell called the sarcoplasmic reticulum. So they are finger-like invaginations of the sarcolemma, extending into the sarcoplasma to surround each myofibril at the region of AI or HI or AI junction. Okay, the T tubules are embraced on either side by the terminal cisternae of the sarcoplasmic reticulum, forming a SRT tubule SR complex called the triad. Okay, so all those words are very confusing, so we'll see the picture lah. Okay, so this is the T tubule. So if you imagine this is the sarcolemma, which covers the muscle fiber. Okay, from the outside, so impulse or the nerves will give information or action potential here at the sarcolemma. So because of the T tubules, the action potential can quickly pass through inside the muscle fiber and be disseminated throughout the whole muscle fiber. Okay, if it's just here, it takes time for the impulse to go through the many layers, right? Okay, that's why the function of the T tubule is to bring the action potential through to all cells at as quick as possible lah, for contraction to happen simultaneously. Okay, so and this is the T tubules function lah. So that's why it's called a T tubule. It looks like a T-shaped structure. Okay. So again here you can see this is the muscle fiber bundle. This is muscle fiber bundle, and these are muscle fibers. Okay, if you see one muscle fiber here, and this is the T tubule. It helps to spread, and this is the neuron. The neuron will give information through the sarcoplasmic reticulum. Okay, and from the sarcoplasmic reticulum into the T tubules, it will disseminate the action potential for contraction of the whole muscle fiber. Okay, so what is the significance of having this T tubule? It, as I mentioned, it rapidly disseminates the depolarization or action potential from the nerves throughout the whole cell or fiber. It results in the release of calcium ions from the sarcoplasmic reticulum into the sarcoplasm, causing contraction. And when depolarization ceases, the calcium is also actively transported back into the sarcoplasmic reticulum via the cisterna, which is via this T tubule complex lah. Okay, and this causes the muscles to relax quickly as well. Okay, in terms of muscle contraction, okay, there are 3 types of muscle contraction. One, the isotonic is divided into 2, the concentric and eccentric contraction, and there's another contraction called the isometric. So in isotonic contraction, length is changed, however the inner tonus is unchanged. So for example here in concentric or contraction here, when you lift the dumbbell, okay, there is a shortening of the muscle, so the length of the muscle is changed. Okay, and in eccentric contraction is the opposite of there's lengthening of the muscle. So in both concentric and eccentric, they are increase in length. Okay, so here there's shortening in concentric, and eccentric there's lengthening of the muscle. However, in isometric contraction, okay, there's no movement occurring, so there's no length change. Oops. However, there is increased workload due to the presence of the dumbbell, and because you want to support the dumbbell in one position, so the tone is still there, meaning there's still some contraction here, but there's no movement, so the length is not changed. So that is called isometric contraction. So there's this, we have another picture here. Okay, so in concentric contraction, for example here, the biceps brachii during the upward phase of exercise, so produces tension and shortens, it becomes short to create this movement lah, which is flexion. Okay, and in eccentric, the biceps brachii during its downward phase of exercise, causing lengthening of the biceps lah. Okay, okay, here in isometric contraction is holding the weight still, there's no movement, so the length stays the same. However, the function is to stop flexion and extension, so there's no movement of extension or flexion, but holding the weight still causes tone to change lah. Okay, and that is isometric contraction, when there's no length change, but tone, there's still increased tone. Eccentric, increased length. Concentric, decreased length. Okay, so you also have abnormal contraction of the muscle in if there is some pathology going on sometimes. Okay, so we can get spasms or involuntary contraction of a muscle. For example, cramps. Cramps are painful spasms. Tetanus is multiple spasms of skeletal muscle. Tick, you can get tick as well. So those are involuntary twitches of muscles. Muscles that are usually under voluntary control, meaning your skeletal muscles. Okay, if they get involuntary twitches, you call them tick. Okay, and tremor, rhythmic involuntary contractions of opposite groups of muscles. Okay, and fasciculations, involuntary short twitches on motor unit visible under the skin. So this is not, you can't really, it's not moving a limb or an organ, but you see the twitches under the skin, like there's something moving inside the skin, and that's fasciculations. Fibrillae are spontaneous contractions of fibers of one muscle that aren't visible under the skin. So fibrillae is abnormal contraction that you cannot see lah. Okay. So, innervation of skeletal muscle. I think I mentioned a bit in the previous lecture regarding motor innervation, autonomic, and also sensory innervation. Okay, so the motor nerves, they branch within the perimysium. at the site of innervation, the axon loses its myelin sheath and forms a junction, the neuromuscular junction, which is the junction, as I mentioned before, between the nervous system or the nerves and the muscle. Okay. Okay, and the synapse here, or the junction here, contains synaptic vesicles with neurotransmitter acetylcholine. Okay, just to show you, so this is the perimysium, remember, so this is where you find your nerves lah. Okay, and these are all your nerves in the perimysium. Okay, so motor unit. What is a motor unit? I mentioned in the previous slide as well, or the previous lecture. So it is a single nerve fiber, and all the muscle fibers it innervates are called one motor unit. Okay, so individual striated muscle fibers do not show graded contraction. They contract all the way or not at all. The fibers within a muscle bundle do not all contract at the same time to vary the force of contraction. Okay, muscles are composed of many motor units, meaning that one axon will innervate many muscle fibers. Okay, the number of motor units and the variable size depends on the intensity of a muscle contraction. So just to show you, this is a motor unit. So one nerve, but it innervates many muscle fibers. Okay, two motor units mean there's two nerves innervating many muscle fibers. Okay, so one motor unit is one nerve and its innervating fibers. So here, if you, for example, we take a section from this biceps again here. For example, so we have 3 motor units. Okay, one in red here, motor unit 1, and one in blue, and one in purple. So basically, just to recap, one motor unit is one nerve with all the muscle fibers it innervates. So if there's many nerves, then there's many motor units lah. So the number of motor unit depends on how many nerves are there. Okay, so the smaller the motor unit, the less force it produces, and the more graded the force production of the entire muscles. Okay, so if we give example to make you understand, so in fine movements, for example, your eyes, okay, they are small, they have many fewer muscle fibers, meaning small muscle fibers. Okay, so they have to be able to contract in tiny amounts so that your eyes can make tiny movements rather than jumping all over as they look around. So these muscles need to have small motor units. Okay, so if you understand, in the eyes, you don't need a great force to contract, because you have tiny muscle units to help with more precise movement. So because you want to look at a precise object at a precise distance using small muscles. Okay, so if in a small motor unit there's less force, so you don't need to like carry a huge object or something like that, so there's less force, but the precision is more. So in coarse movement, say your quadriceps muscle, they are huge, they contain a lot of muscle fibers. They need to produce a lot more force as well, because they help your body to support your body when standing up lah. Okay, so they have large motor units because they need more force. Okay, and it won't really cause problems if they are not precise in movement, because they just help you to stand up or walk. So you don't need to actually like think about walking in more precision and so on lah. Okay, so as for small motor unit is for fine movements and more precise movements with less force needed. In coarse movements, you have larger motor units with that need more force but less precision. Okay. So sensory receptors, I mentioned the muscle spindles just now. They help with proprioception. where you understand where your body is positioned at, right? So when you close your eyes, you still know where what your hands are doing, because you have this sensory receptors called muscle spindles that give you information regarding the position of your body. Okay, even when you cannot see the body parts lah. Okay, so this is called proprioception. So so muscle spindles are found more densely in areas that need fine control. Example, in the hands, because in the hands, for example, when you write, you need fine control, right? So you need a lot of feedback on the proprioception or the sensory, so that you can move your hand accordingly when you write and do precise movements with your hands, for example. Okay. So here, this is a muscle. Okay. So if you cut through the muscle, you will find a muscle spindle in between. Okay, usually it is dispersed inside the muscles lah. Okay, and you have these afferent nerve fibers supplying the afferent means sensory, okay, that give sensory input. Okay, at the tendon as well, you have one Golgi tendon organ with an afferent nerve fibers, and inside the muscle you have the intrafusal muscle fibers, or the that makes up the muscle spindle. Okay, the muscle spindle is the whole thing. The intrafusal muscle fibers are the muscle fibers inside the muscle spindle lah. Okay, the extrafusal muscle fiber is just your normal skeletal muscle fiber lah. Okay, and you also have stretch receptors throughout or surrounding your intrafusal muscle fibers, forming the muscle spindle. So it is made of this capsule, the intrafusal muscle fibers, and your stretch receptors, giving information through this afferent nerve fiber lah. So during movement of the muscle, both your muscle spindle and Golgi tendon organ will give afferent or sensory information to the brain, and then later from the brain will give order to your muscles to control the movement according to this sensory information lah. Okay. Okay, so just a bit on Golgi tendon organ. So it's the same function, which it is for sensory information, but the sensory is in terms of tension in the tendon lah, instead of warmth and so on. It based on the tension of the tendon, or the here kan, the tendon holds the muscle, so when the muscle moves, it will create tension in the tendon. Okay, and this tense motion of the tendon will give information through the Golgi tendon organ lah. Okay, so I've mentioned about isometric contraction and isotonic contraction, right? Okay, so cardiac muscle structures. Okay, so that's skeletal muscle. All this is in skeletal muscle fibers. Okay, so we'll talk a bit about the cardiac muscle now. So in cardiac muscle, it's a different organization than the skeletal muscle. Okay, it has branching of the fibers. Okay, it's not as closely tied or packed as the skeletal muscle just now, and it has these red or purple dark bands here called the intercalated discs. Okay, when you see intercalated discs, you know you're looking at cardiac muscle rather than skeletal muscle. Okay, because these intercalated discs create junction for action potential to be shared among all the cells of the cardiac muscle. Okay, cardiac muscle also has troponin, which is a contractile protein found only in the cardiac muscle lah. Okay, specific to the cardiac myocyte. So each So troponin originates within the sarcomere of both cardiac and skeletal muscle. Each sarcomere is comprised of actin and myosin filaments which interact to produce a muscle contraction. Okay, while troponin is found in both skeletal and cardiac muscle, the high specificity of CTN for cardiac myocyte necrosis is conferred by unique peptide sequences present in CTN but absent in skeletal troponin. So meaning there are three types of troponin, T, I and C. So T and I are more specific for the heart muscles. However, C can be found anywhere in the body. Okay, so if there is myocardial injury, so patients who have myocardial infarction or heart attack, as you normally refer it to, in heart attack, the cardiomyocytes are died off, or they become injured. Okay, and because of this injury, they will release this cardiac troponins, which are T and I, into the blood. So when this happens, when you do a blood test in the patient after the heart attack or myocardial infarction, you will find high concentrations of cardiac troponins T and I. Normally, if you do a blood test on a person who doesn't have heart attack, they will not find these cardiac troponins because they are not found in the blood in the serum. But because of the death of the heart muscle cells, they are released into the blood. So that's why you find it in those who have had a heart attack So the last type of muscle is the smooth muscle. Okay, they are elongated spindle shape. They have no striations. They have a single nucleus in the central position and is surrounded by viscera. Function mainly in involuntary control, as I mentioned earlier. So your GI tract for peristalsis and so on. So blood circulation, so all your blood vessels are lined by smooth muscle, secretion, excretion, and so on lah. Okay. So this is an example of your smooth muscle. It is spindle shaped and with one nucleus in the center. So here, spindle shape with one nucleus in the center. Okay, in your practicals, we will go more into detail regarding the microscopic view of the different cells. Okay, so just to recap the three cells, so we have your skeletal muscles, okay, which are long cells or fibers with many striations, and we call these striations. Okay, and contraction is for voluntary movements. So all your skeletal muscles that move your bones. Okay, so these are your skeletal muscles, and they have multiple nuclei at the periphery. However, smooth muscles, they are spindle shaped. Okay, long spindle shaped cells with one single cell in the center. Okay, usually we find them in GIT or in the blood circulation, the vessels. Okay, they help with propulsion of substance along internal passageways. Next is the cardiac muscle. Okay, you have striations, you have these lines, striations, and you have branching. Okay, in skeletal muscle you don't have branching. Here you have branching of the muscles. Okay, and you have these intercalated discs, which is important to create synchronized contraction in the heart. Okay, so location wall of the heart, it helps with pumping of the blood into the circulatory system. Okay, so these are the three types of muscle that you can find in the body. Okay, thank you very much. Oh, can you take the attendance? Can someone share the link for attendance?
Doctor, I have a question if I may.
Okay.
Will we have the assessment, the mid-course assessment on this Friday online?
Oh yes, Yes, I forgot. Yeah, you will have the assessment online this Friday. And the topics that will be asked will be any topics before the assessment itself. So in the timetable, anything that you have learned before Friday, we can ask you.
All right. Thank you very much.
Okay. And it's a single best answer. So there's no, it's not written assessment lah. It's just A, B, C, D, multiple choice.
Doctor, is it
Yes.
this week Friday or last week Friday?
Pardon?
You say the topics are until Friday. Is it this week Friday or last week Friday?
Oh, this week, of course.
Oh, this
Last
week Friday means like all topics will be Monday, Tuesday, Wednesday, Thursday will be asked. Is it?
Not will be, but can be.
Oh. Okay.
Okay. So, and we have
Uh,
practicals on Wednesday.
Doctor, how many questions are there going to be?
How much? 30 questions.
30 questions.
Usually we have 30, but I'm not sure for online are we going to increase it or not, but most likely we'll stick to 30 lah. I have to discuss with my committee today. We'll have a meeting. I will inform you after that lah. The latest, because we were just informed about the online class, online assessment. Previously they said we were going to do it later, like postpone it after CMCO, right? But then they just told us that we have to do online. So we will meet today and discuss. But most likely it won't change lah. I think we will stick to 30 for now. Okay?
Thank you, Doctor. Yeah.
Okay. Okay, remember to do in the attendance, you need to do it for both classes, GA10 and 11. Okay, so I'll see you on Wednesday for our practical, inshallah.